Okay, so I have a kind of broad talk here, and you can derail it and ask questions if you like, or we can follow the thread, whatever's of use to you guys. And I want to start by pointing out how much progress we've made in my field in the last 20 plus years. And that's easiest to see by stepping back and asking what we knew about the functional organization of the human brain around 1990. So here's my kind of dopey schematic of the state of knowledge, 1990. And the black ovals are primary sensory and motor regions. Their locations were known in the human brain for 100 years before that. And the big colored blobs are mental functions like face recognition, language understanding, and attention, where there was a bunch of evidence, even back in 1990, and had been for a long time even then, that if you had damage to those approximate regions of the brain, you would have problems with these mental functions, right? But you can see that those blobs are big and blurry, and there are not very many of them, and there basically was just like not a whole hell of a lot known about localization of mental functions in particular regions of the cortex. And importantly, back then, it wasn't clear that there was much more to be known. That is, it wasn't clear that if we had better methods to look at functions of different patches of the brain, that there would be a lot more fine-grained mapping of particular mental functions to particular brain regions, right? Maybe that was it, right? And then functional MRI came along, and here's where we are today. So this is, again, very schematic. There aren't neat little colored ovals in the brain. Everything's muckier than that. But what the diagram is meant to show is that there are now dozens of regions of the cortex in humans where we have a pretty good idea of the function of what goes on in that region of the cortex. Okay? And so what I want to do is take a moment to give you some ancient history on what exactly it means to say, like, what do I mean when I say that little patch does face processing? Okay, so I'm going to tell you about some really old experiments briefly, and then we're going to move to the cutting edge where the field is now. But I want you to have a sense of what I mean by making little colored blobs on the brain. Okay, so let's take this face patch here. What do we mean by saying that that region is engaged in face processing? So about 20 years ago, we did the following incredibly simple-minded experiment. Pop people in an MRI scanner, show them pictures of faces like those and objects like those, and just have them look at the stuff and scan their brains. That's it. Pretty rudimentary. Then for each little three-dimensional pixel or voxel in their brain, which, by the way, contains about 500,000 neurons, so our resolution is the best you can get in humans, non-invasively, but it's appallingly bad compared to any of the stuff you guys are doing with animals, right? And so you ask of each little 3D pixel, each little bit of 500,000 neurons, whether the neural activity measured with functional MRI, very indirectly, but that's best we can do, whether that neural activity is higher when the person's looking at faces than when they're looking at objects, okay? So if there's a bit of brain that's specialized for face perception, wouldn't you think it would do more when you serve up faces than when you serve up telephones and ears of corn and crabs, right? So that's the idea. And so when you do that, you find that indeed there's a little patch of the brain on the bottom surface of the right hemisphere, approximately in there, where a little cluster of voxels, a little patch of brain, where the stats tell you that those voxels are producing a higher MRI signal when the person was looking at faces than objects, okay? And so whenever you see brain images with nice little colored blobs, you should be suspicious. There's lots of ways to get colored blobs when you don't have the real goods. And one way to tell if somebody has the real goods is ask to see their raw data. So here's mine. So this shows you the time course of the MR signal coming from that patch of the brain over a 5-minute scan when the subject was staring at a dot, looking at faces, staring at a dot, looking at objects, and so forth. Okay, can everybody see that, you know, by the eyeball test, that bit of brain responds a whole lot more to faces than objects? You can also see it responds more when the subject is looking at objects than when they're staring at a dot. So when I say that a patch of brain is selective, I mean it responds a whole bunch more for X than anything else. I don't mean it responds 0 to everything else, okay? Make sense? Everybody on board with this? Okay. So that's the bare basic step one. And you know, as you can see here, it's a big effect. And we see something more or less like that in pretty much every normal person we scan, okay? The exact location varies a little bit, but it's always, you know, back in there someplace on the lateral side of the fusiform gyrus in the right hemisphere. So now the question is, does this tell us that that region is really selectively responsive to faces? What do you guys think? Are we done? Have we nailed it? No, why not? You in the blue t-shirt who's trying to eat your lunch, sorry. Good. Give me another hypothesis. What else might it be? This is how you critically evaluate a claim. I'm showing you these data, and I'm saying, therefore, that bit of brain is selectively involved in face perception. The way you evaluate that is you think, what else might be true instead, given those data? So give me some hypotheses, starting with you.
It could be selective for features that are more common in faces than other objects.
Yep, like? Could be just hair. There's a hypothesis. Not faces, just hair. Okay, what else? Emotion? Yeah. We're social primates. We get all kinds of other stuff from faces. Maybe it's the emotional content of the faces. These were kind of unexpressive, but you could still read in emotions. What else?
Just looking at people?
Yes, people. Maybe not just faces per se. Maybe people. Maybe it will respond to bodies, right? So this is the crux of testing a hypothesis. You start with some data, and then you consider all the alternatives like that. Okay, so me in my field, we've been doing that for 20 years. I mean, I sort of got sick of it about 10 years ago, but we did that for a long time. Because I like to take these hypotheses seriously, not just toss them out there and say, maybe X, on to the next thing. It's like, okay, maybe X, let's find out, okay? And so here's an example of how you can drill down, take those alternative hypotheses seriously, and test at least some of them. Okay. So these are some of the things you guys came up with. Does it respond to anything human? One of you said that. Any body part, anything interesting, anything curvy, and blah, 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 right? It goes on and on. Okay, so here's an experiment we did also way back in our first paper. We said, okay, let's try different kinds of faces. Oh, and hair, right? I didn't list that, but hair. Let's hide the hair, okay? And let's have 3/4 views. And let's compare the response to faces to the response to hands, another human body part that's kind of curvy, has flesh, all that stuff. And let's further make subjects pay more attention to the hands and the faces. We do that by having subjects do a one-back task. That means they press a button whenever 2 consecutive images are identical. And that task is easy for the faces and really hard for the hands. Okay? And so we basically do the same experiment in the same subjects. Same subjects who do this, now we have them do that experiment. Okay? Alternating periods of 20 seconds of this, 20 seconds of that, blah, blah. Okay? I'm trying to skip over stuff because I think this is really old, but I want you to get a concrete idea. So if any of it isn't clear, ask me a question. Okay, so what do we get in that contrast? We get the same blob in the same person, pretty much exactly. And if you look at the time course of data, that blob goes, oh, actually, this time course comes from those exact voxels. Okay, so nail them down here. This is our candidate face area. Let's test it. And yes, it responds much more to faces than hands, even in this condition. And so with one simple little experiment, we have definitely knocked out a whole bunch of hypotheses. It's not just anything human. It's not just any body part. It's not anything you pay attention to. It's not anything curvy by some loose definition of curvy, and so forth. It's not just hair, okay? So that's just an example to give you the gist of how you can take a hypothesis seriously, take the alternative accounts seriously, test them, and knock them down, okay? And so I won't go through all the details, but me and lots of other people spend a lot of time doing that. If you haven't seen this image before, raise your hand if you can tell what it is. This one right here. Okay? Raise your hand if you can tell what that one is. Right there. Come on, you guys, you can do it. Can't tell? Yeah, you're being shy here. If you haven't seen it, don't worry, it's nothing wrong with you. It's a profile view of a face, eyes, nose, mouth. Okay? After you've seen a few of these, you start to pick up on it and you see them. But after you've seen a few of those, you don't pick up on them. You can't do it. Even when I tell you now, that is the same image upside down. It's a face, it's upside down. Try seeing the face in that. Best of luck to you, right? Okay, so that's a nice control. I'm an atheist, but I think inverted faces are God's gift to the face researcher, right? Because all of these hardcore vision people who want to say, oh, Kanwisher, your face region isn't selected for faces, it's selected for this kind of spatial frequency and that kind of mathematical gobbledygook, blah, 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 blah. I can say, no, same stimulus. All I did was turn it upside down, and the response magnitude goes down by almost a half, right? Corresponding to the fact that people see the face usually when it's right side up, and they never see the face when it's inverted. Okay, that's just another example of how you can knock down alternative accounts. Okay, so the bottom line of all of this is, if you do this long enough, you can really make a pretty strong claim that that region responds much more to faces than anything else. Okay, so that's mostly what I mean when I put a little colored blob on the brain. I mean, we've gone through a whole rigmarole like this, considering all the alternative accounts that seem remotely plausible, knocking them down, and we do all of that before we stick a colored blob on the brain. Okay, and so these kinds of things have been found across lots of different kinds of contrast, lots of different kinds of faces, lots of different kinds of non-faces, lots of labs all over the world, lots of methods. It's pretty solid. Okay? Okay, so that's what I mean by sticking a colored blob on the brain. And as I mentioned, like, most of these things are widely replicated by many people, and the selectivities are really strong. Like, I'm not that interested if you find some part of the brain, you know, that responds like this to X and like that to Y. If you run 100 subjects and that's significant, well, who knows? Like, I like things with their big effects. Feels to me like when you get a really big difference, nature's trying to tell you something. Yeah? Okay, so these are all things where they're really big effects. Okay? Okay, so I'll just mention a couple. I'm not going to mention. I had two little other points, but I'm going to skip them. Two particularly cool regions here, but there's other things I want to get to. So these things shown here are present in approximately the same location in pretty much any normal subject. We could bring any of you into the lab, and if you spent, maybe 2 scanning sessions, total of 4 hours, we could find all these regions and map them all in your brain, and they'd all be more or less like that. A little rattier, because real biology is messier than this, but basically recognizably like this. Yeah? And I think this is incredibly cool, because I think this is a kind of initial rough sketch of who we are as thinkers. I see this as a diagram of the human mind. Here are the basic pieces of cognition that we can see by looking at the brain. That's what I'm in it for. Yeah? And I think that's really important progress, especially compared to where we were in 1990. But it is also the barest beginning. Like being able to say, this little patch really, really mostly does face recognition, and this little patch really, really mostly does language understanding. That's cool, it's important, it's worth knowing, but it's rudimentary as hell, right? It's just so basic, right? So what I really want to do in this talk is show you what I see is the sense in which this diagram is really a diagram of a future research program, of all the questions it raises and some of the little snippets of how one can go at addressing them. Okay, so you can see it as a roadmap of research, this huge set of questions that it lays bare, and I'll just start marching through a few of those. Sound good? Everybody with me so far? All right. So one of the first things you might say, looking at this diagram, is, really? These regions just do one thing? I should say that most of my colleagues have this reaction. Like, they don't really believe this, right? They think I'm getting carried away with my claims about specificity. So I'm going to show you a shred of data. Oh, actually, first I'm going to give you the strongest counterargument. So sometimes it's just like skepticism. It doesn't seem right. And sometimes it's like arguments that people have raised that have since been refuted. But there's one very smart, serious piece of counter data that I take very seriously that argues against this specificity that I want to tell you about. And that is, I didn't put enough of diagram in here, but I'll just like act it out. So all of the stuff that I've been talking about so far is the most basic kind of analysis you can do with functional MRI. Ask of each voxel, is a signal higher in this condition than that condition? Very rudimentary. But you can come back and say, okay, here's this face area. It's, depending on your resolution, maybe 100 voxels in the brain. Little patch of brain about 1/2 centimeter across. There it is, right? And so you might say, look, even independent of the overall magnitude of response, how much neurons are firing on average over that whole region, maybe there's information in the fine-grained pattern of response across voxels in there, right? And so there's a whole method that's been around for about 10, 15 years called multiple voxel pattern analysis, in which you look at the fine-grained spatial pattern of response across those voxels in that region, and you ask what information is contained in that pattern, right? So for example, if this is the face area right here, and we scan a bunch of people, or we scan one person, let's say, and we find that when they look at cars, the response to faces is up here. The response to cars is really low, but there's some systematic spatial profile. When they look at shoes, it's also really low, but there's a different spatial profile. That means, if the data are strong enough, that you could look at the pattern of response in the face area and tell if the person was looking at cars versus shoes. Two things, neither of which are faces, okay? And if we think, as most people do, that the brain is an information processing device, then it's information we should care about, not activation. And so this is a very, very serious, it's one, a really important method, and two, a very important challenge, initially raised by Jim Haxby in a paper in 2001, where he argued that if you look inside the face area, you can tell from the pattern of response if people are looking at, I forget if it was cars versus shoes or scissors or what, a bunch of different things, right? And at first, I tried doing the same experiment. I said, well, you know, you did it wrong, and when I do it right, no, you can't. And then a couple of years later, we did it again more carefully. It's like, oh, yeah, you can, a little bit. It's significant, it's crappy, but you can tell, right? And then Jim Haxby published a paper saying, like, yes, you can tell, but actually it's really, really weak information. And if you want to know about cars or shoes or anything else, that's the last place you'd look. So we've sort of converged on exactly agreeing that actually, yes, there is information, and it's really weak, but it's significant about non-faces, right? So this is important if you're exercised as I am about the idea of extreme specificity in the brain, because it's the strongest, smartest piece of counter evidence. Did everybody get that more or less? I was a little vague. Okay, yeah.
So in terms of the areas themselves, like, we have a lot of neurons. So would you say that that is like what we can, according to technology, nail down as the smallest unit of like a whole cognitive process, or if electrophys and other animals show anything, or even humans, like that it's more a couple of neurons, like much like an order or two magnitude?
Yeah, this level of organization there is just what we can do in humans, right? And so if you have a monkey, you know, many of these things in the bottom of the brain, after we first reported them in humans, the monkey guys went and looked for them in monkeys and found them in monkeys. And then because they were monkeys, they stuck in electrodes and they recorded from lots and lots of neurons in those regions. And they have magnificent data showing the actual neural code for all of these things that you can see unit by unit over hundreds of neurons, over hundreds of stimuli in monkeys. It's magnificent. And sad indeed that we can't do anything like that in humans, right? So there's much, much finer grained information available in animals than there is in humans. My lab is actually not doing very much work on face recognition anymore because actually I think the face system is extremely similar in humans and monkeys, and probably homologous, that is inherited from a common ancestor. And I think, and the methods are vastly more powerful in monkeys. And so I think, you know, there's a few things you still want to do about faces in humans, but mostly you want to really understand the neural mechanisms underlying face recognition. You do it in monkeys. Now, luckily, that's not true for everything on this diagram. That argument doesn't work for, say, language understanding or music, right? Or lots of other things. But yeah, I think the grain of information is vastly sharper than this, and you can see that in animals, mostly not in humans. Yeah.
So as far as like the point of this counter argument, are they saying that this like isn't a face area at all, and that specificity isn't there because that faces are easier to detect, or is it saying, yeah, this is a face area, but it can also do other things?
Yeah, more the latter. You could take it different ways. But the point is, if there's information there about things that aren't faces, that is a strong challenge to at least the most extreme view of face specificity. Right, and I'm focusing on faces just to be clear, but the same is said about a bunch of these other regions as well. Okay. Okay. So this is a smart counter argument. So let's see if we think it's really true. I can respond to this counter argument and say, okay, I agree now. There is information in there, but the fact that we scientists can look at that pattern and read out a little bit whether the person was looking at shoes or cars doesn't mean that the rest of that brain is reading that information out of there, right? And so now we get to a really deep question of what is the causal role of that pattern? Does it have any causal role in behavior, right? And so in science, we want to be testing causal theories. We don't want to just say, when this happens, that happens. We want, oh, did this cause that? Did that cause that? Were they both caused by something else? We really want to know. That's the essence of scientific understanding. And so we have very few causal methods in humans. And so we're usually having to very indirectly infer causal structure, but it's of the essence in science to look at causal structure. And I think it's particularly important in this case because you might worry that these shrimpy little patterns don't necessarily mean, aren't necessarily playing any causal role. For example, if you designed a face processing system and you fed it shoes and cars, you'd probably get different patterns out the top. So the very fact that you can get different patterns out doesn't mean that that's what it was designed for, developmentally, or what it's being used for in that brain, right? So it's really worth testing the causal model. Okay, that was a lot of preamble to show you my favorite video. So we got this opportunity to test this with this very generous gentleman here who has a lot of wires all over his brain because he's being mapped out before neurosurgery. He had intractable epilepsy, and what neurosurgeons often do if the epilepsy does not respond to drugs, which it usually does but sometimes doesn't, when it doesn't, sometimes the best thing you can do is find the source of those seizures and surgically remove it. And sometimes what neurosurgeons do before that is take out a flap of bone, it's totally gross, stick electrodes all over the brain, and have that patient hang out in the hospital for a week or so, waiting for them to have seizures, so that each time they have a seizure, you can figure out what the source is. And also, so that you can map out functions while those electrodes are sitting there, so that you know what to steer around if you have to cut, okay? So this guy, here's a map of the bottom of his brain with all the electrodes that the neurosurgeons had put in. They called me the day after they put them in and they said, do you want to collaborate? We're going to be recording responses from these electrodes and stimulating them electrically. It's like, damn straight, I want to collaborate. And so just for comparison, here's the bottom of my brain. That's my face area in red. And the purple bits are bits that respond more to color than grayscale images. They're not as selective as the face areas, but they're preferentially responsive. And so you can see that those electrodes seem to pass right over lots of good stuff, right? And so the first thing we did was to send a bunch of stimuli to Japan where this guy was being tested, and they shipped back the responses. Now we're just talking electrical responses measured directly from the surface of the brain. This is very, very rare special data, the kind of thing we almost never have in humans, and when we do, we're just like blown away, right? And so this little electrode right there that looks like it's sort of in the same position as my fusiform face area, here's its response. We had a bunch of different stimuli, bodies, faces, digits, 2 different kinds of Japanese characters, line drawings of objects, and photographs of objects. And this is the magnitude of response of that electrode, and you can see it's damned face selective. Like, I said before, with functional MRI, you get a little bit of a response to objects in the face area. Here's like nothing, pretty much, except for bodies a little bit. Okay, yeah?
Is that ERP, or is that like spiking?
Okay, these electrodes are about 2 millimeters across. They're sitting subdurally right on the surface of the cortex. So they're not single units, but they're also not ERPs. This is actually power in the high gamma range. And so the people who have been doing this kind of work in humans have shown that raw ERPs from the surface of the brain, sometimes you can get face selective responses. Those have actually been reported way back. But you get much clearer, so first of all, the high gamma power, which is like, 60 hertz and above, power in that frequency range is most correlated with spike rate when you have a chance to measure both. We don't have a chance to measure both here, so this is high gamma, okay? Yeah?
Why did you choose the 2 different reading units, or like the 2 different writing systems?
I can't exactly remember. We put this together on it's like we got the phone call, we made the stimuli, they did the experiment the next day in Japan. We're like, okay. There's a visual word selective region in the left fusiform gyrus that looks like those electrodes might have gone right over the top of it. And we just thought if we got that, 1, that would be cool, and 2, it would be interesting if the phonetic versus other writing systems, produced a differential response. Yeah. Okay, so everybody get this? So this is basically validating with a much more directly neural measure that we rarely get in humans what we had seen before with functional MRI. Okay, so that's nice. It's not earth-shattering, but it's nice, very nice to replicate stuff with a different method. Right next door is an electrode that does this. So the outer lines are the version of that stimulus when we present it in color. And the inner lines are the response to the version of that stimulus when we present it in grayscale. And so you can see that that electrode, responds a little more to the characters and digits than other things, but basically is not very selective for the stimulus category, but for each category it responds more when it's in color than grayscale. Okay? So again, we're just replicating stuff we've seen before with functional MRI. Okay? But the whole reason I'm going through all this rigmarole is that we then got to do the causal test, because the neurosurgeons had decided to electrically stimulate those electrodes. And so I'm going to show you a little video of what happens to this guy when they stimulate his right fusiform face area. Okay, it's in Japanese and it's kind of quiet, but there's subtitles. So here we go. So he's looking at a face and he's getting stimulated. He doesn't even know there's a face area, let alone knowing what electrode's being stimulated. I don't know what's going on. Your eyes change. Not here. He's such a great subject.
One more time.
Okay, that shows the causal role of that region in face perception. But what I really wanted to know is, does it have a causal role in the perception of things that aren't faces? So he's looking at a box and getting zapped, and he says,
No change.
最初に1秒ぐらい始まって1秒ぐらいが横に目口って感じでちょっと一瞬見えたんですけど、これ何かなと思ってたら気がついたらもうこの箱にしか見えないっていうか。
You know, he's looking at a soccer ball.
なんですか、またさっきの横に目と口って感じで違う。
Eyes.
This is a kanji character. It's on a card that's foreshortened.
More, please. One more time, please.
やっぱり顔に見えました。目、口らへんって感じ。
Okay, so the point is, one, that region is causally involved in face perception, for real, not just turning on when you look at faces. And two, it's not causally involved in the perception of things that aren't faces. In fact, all it does to things that aren't faces is plunk a face on top of them, right? So we replicated this multiple times. I'll show you a little bit more of data in a second, but I think this is a strong counter suggesting that, okay, there may be like weak little patterns about other things, but those weak little patterns aren't doing very much because you can like really blitz them and nothing happens to the percept of things that aren't faces. But you might worry, maybe any place you zap in the brain you see faces. We're social primates, we like faces, we see them in clouds, maybe you go like that, you see a face. So let's ask what happens when you stimulate that next door region that responds more to color than grayscale. Okay? Okay, he's looking at the box. getting zapped right next door. Again, he has no idea where he's getting zapped. He doesn't know there are color preferring patches of cortex. And he says,
この箱の真ん中見てたら、この半分ぐらいですね。なんか虹色になんか光ったりしてますね。この真ん中見てたら。ボール見てたのもやっぱさっきの明らかに幅広い感じ。なんかちょっと虹色に点滅してるんですけど。先生の鼻あたりに目線見てたらやっぱこちらの顔半分がまあ同じような感じで半分が
yeah, only 2 adjacent electrodes, right, in that one spot. Yep. Okay, so what that shows is there's a very precise mapping between the regions that respond selectively to X and the percept you produce when you stimulate it. And that shows the extreme specificity of the causal role of these regions. And I think it's at least a decent first pass response to the very important critique from pattern analysis. And it further suggests that if you have to guess about the function of a region, don't mess around with fine-grained pattern analysis. Look for big selective responses. Yeah. Okay, so that was all the first question. We're totally going to run out of time. But let me just say a few things to clarify. So my basic claim is that many of these regions really do pretty much just one thing, right? You know, some of them there's like a little bit of a niggle, maybe just a little bit of this, a little bit of that, maybe kind of, but they're really selective, okay? But importantly, I don't think that's true of the whole cortex. In fact, we know it's not true of the whole cortex. There are big swaths of cortex, like for example, these regions that I've circled in gray there, that are well known to do all kinds of different stuff. So we have experiments where we can show that the very same voxels in those regions are engaged when you do 7 totally different difficult tasks. What those regions seem to be most interested in, how much mental effort you exert in doing a task. They don't really care what the task is. So they're sort of like the opposite of all the little colored blobs. So I just want to make it clear that I don't think that every little patch of cortex does just one thing. I think some patches of cortex do just one thing. Okay? Second, this claim about functional specificity, that some patches of cortex do just one thing, it's important to distinguish that claim from a bunch of other ideas that my colleagues sometimes confuse it with. You guys wouldn't confuse them with these other ideas, but let's just make sure everybody has it straight. So first of all, just because some patch of brain responds selectively to something doesn't at all mean that that region of brain is innately specified for that function. That's a deeply interesting question. For most parts of the brain, we have no idea and no good way to tell. But it's just a totally different question from the specificity you see in adulthood, right? And there's at least one case where we know that there's a very functionally specific patch of cortex where the selectivity of that patch had to be learned and can't be innate. And that's that little visual word form area that I briefly mentioned before, the bit that responds selectively to visually presented words and letters. And we know that region is learned from experience because humans have only been reading for a few thousand years. And that's not enough for natural selection to have built in an innately specified, specialized piece of machinery for reading. And further, it responds selectively to orthographies you know, not orthographies you don't. And further, it develops right after you learn to read, not before. All of those things nail the case that that bit, at least, is learned. All the other ones we basically don't know. Yeah? Okay, second thing is, I'm going to skip these, or not. Okay, I'll mention that one. The claim that some little patch of brain is selective for some mental function in no way it should be taken to imply that that piece of brain functions independently of the rest of the brain. Every bit of brain needs to get input from somewhere else, or it has nothing to process. It has to send output to lots of other regions, or there's no point in processing it. Nobody will know what it finds. And probably it has lots and lots of other online interactions with other regions as it processes. So a lot of my colleagues like to say, but the brain is a network. It's like, duh, of course it's a network. In no way does the network property of the brain argue against the specificity of some patch of cortex. Yeah? Yeah.
So my question is, so does it interact with like some does it more so like depend on your experiences and what you witness in your life? Or
Yes, everything will, right? So you have some processing pathway coming up your visual system, and we're targeting a reasonably high level stage in that processing pathway with these electrodes, which is why we're getting meaningful stuff out rather than just flashes of light or something. And of course, that processing pathway continues. Like when you go around in the world and look at stuff and know what you're looking at, the reason you know what you're looking at is you've learned what those things are, right? So when I ask about innateness, I'm not talking about innateness of, my knowledge of what mandana looks like. That couldn't possibly be innate. I obviously learned that, right? And so did you guys, right? What I'm talking about is it innate that a patch of brain is going to be selective for faces in particular? So lots of the ways that all of these things operate are going to have to be learned, and at some point we have to make contact with stored knowledge. It's a deeply frustrating to me and kind of embarrassing fact that nobody knows where our stored knowledge of familiar faces lives in the brain. Like, isn't that shocking and lame, actually? But nobody knows where it is. It's probably not, I don't think they're all stored right within the fusiform face area. I think they're probably someplace else, but we don't know yet. Yeah?
On the topic of innatedness, when you're trying to look, like in the visual system particularly, when you're doing any of the hypotheses behind like ensemble statistics, like is that something that we do innately, or is that something that's learned, kind of like parsing a scene or something like that, or like parsing a face? Or, like,
You got to say more what you mean, ensemble statistics, right? What you want to ask.
so like if you're looking at like the whole room, like is that something that you learn, like to look in different places and then assemble an image, or is that something that's innate?
I think a lot of that we don't know. The best stuff we know in that space comes from behavioral studies in infants, where you can show that infants as young as 3 months understand a lot of what they're looking at. They understand not just that an object, you show them an object and it hides behind here. They know that object is continuing to exist. They know this object can't move through that object. They even know if you show them social animation stimuli of two little shapes that are kind of socially interacting with each other. This one's chasing that one or flirting with it, versus this one is hitting that one or harming it. They can tell. 3-month-old infants know the difference between a cooperative interaction versus a competitive interaction. So very young infants can perceive all kinds of rich stuff from visual stimuli. It's very hard to go from that, though, to say, can you learn that in 3 months of crappy vision that infants have? That's tough. That's really tough. I think those questions are ultimately going to be answered in animals because you can control experience, and those experiments are being done now in animals. Really nailing innateness questions in humans is damned hard. We're trying to do a study now in blind subjects to see whether when blind people feel a 3D printout of a head with a face on it, do you get activation in the fusiform face area? I'm not sure yet, but that might get close. But it's really, really hard to address questions of innateness in any aspect of vision in humans. But I'll just earmark a vast and fabulous literature on controlled rearing. So there's a guy in Italy named Valortagara who studies chicks, baby chickens. There's a chicken farm nearby, and so they have this wonderful system where they get fertilized eggs. They bring them into the lab. They hatch them in the lab in a situation where they can precisely control their visual experience. Then they do a single trial experiment, like can the chick tell X from Y? And then they send them back to the chicken farm. Isn't that great? Anyway, and scientifically it's really deep because they show all kinds of perceptual abilities that those chicks have on the very first trial that they have ever seen that thing, which they know because they've controlled their experience since hatching. But that kind of stuff with humans, mostly forget it, unfortunately. Yeah?
Is there a mathematical way to say, like, if I'm able to linearly decode some information from one part of a brain, that it's being used by, like, an upstream brain region? Or is there, like, work being done on that? Does that question make sense?
Wait, the fact that you can decode it from some region
Doesn't necessarily mean you can...
doesn't mean anybody's using that.
Yeah, so is there a mathematical method to, like, provide evidence that this upstream
Okay, so Rebecca Sachs and her postdoc Stefano Anzalotti published a paper last year, maybe just on bioRxiv, Anzalotti, where they are trying to develop a method to look at that. It's very impressive and very cool, but it involves a lot of mathematical rigmarole with tiny effect sizes. So, you know, bless them for even trying it, but I don't know. See what you think. And in animals, there's loads of things you can do to test that. Yeah. Okay, so all of this was question 1. Sorry about that. I don't know what to do here. I'll just go on and you guys can ask me, you know, I don't, should we stop and have you ask me questions or should I keep going? I'll keep going. All right. Another obvious question you might think of when you look at this is like, okay, that's weird and cool, but what else? What other crazy stuff is lurking in there, right? I'll just give you the gist of, I don't think, in fact, it's pretty clear that we do not have a specialized bit of cortex for every important thing you do, right? There are lots of important things you do that for whatever, I actually think it's a deep question and I don't know the answer. I don't think anybody knows the answer. Some important things we do can cohabit with other functions in the same brain region or can cohabit with lots and lots of other functions in the same brain region. And some mental functions get their own private patch. And God knows why. I think that's extremely interesting. And I think someday maybe computational modeling will give us some purchase on understanding why. But right now it's just kind of empirical. But beyond these, I'll mention a few other cool ones that I'm excited about. So a few years ago, Sam Norman-Hegner, working with me and Josh McDermott, found a little batch of neurons sitting right up approximately there that respond extremely selectively to music. And so here's a little shred of our data. This is a response of that neural population to 165 different sounds of all different kinds of sounds, dogs barking, toilets flushing, people talking, music of 30 different kinds of music that we played to subjects in the scanner. And that little batch of neurons responds, this is its response profile, and you can see all these high things are instrumental and vocal music, right? And even like this first orange one that's like mechanical sounds, if you actually say, okay, what is that mechanical sound? I think that one is the Mac chime, you know, da 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 da, right? Well, it's like, okay, is that music? It's close, right? So anyway, so this is cool because nobody even knows why humans have music in the first place, right? So all human cultures who've been studied have music, and people have been puzzling about this for hundreds of years. Darwin wrote all about how, you know, it's obvious why humans have language. There's great adaptive value in sharing information with your conspecifics. And so it makes perfect sense to have language and possibly to have innate mental structures that enable you to have language. But music, what adaptive value does music have? And so it's a big puzzle. One of the stories people have told for a long time is that we have music because we can take that neural mental machinery we have for language, which is more understandable why we have it, but once we have that, we can muck with it and get it to do other things and do music with it. And our data say, no, that's not the case, because the response to English speech and foreign speech are the green colors way down here. This neural population that responds like crazy to all different kinds of music doesn't respond much at all to speech. And in fact, other nearby regions that respond strongly to speech respond pretty much not at all to music. So it's not the case that music is co-opting mechanisms we have for speech and language. It's its own separate thing in the brain, which is pretty cool. Was there a question that I barged over? Okay, all right. So that's one cool new thing. Another thing that we have started working on recently is this question of how we understand intuitively the physics of the world around us. And it's something that is kind of easy to not realize, but if you think about it, every time you make an action, if I pick up this can, if I turn to go out the door, if I go to pick up the Mac, in each case, I have to have a model in my head of the physics of the world I'm acting on. I need to know how heavy that object is before I plan the grip that I will use to pick it up. I need to know how heavy the Mac is. I need to know that I can walk over here, that the floor will support me. And that if I want to get out the door here, I can't go through the wall there. I have to go through that opening, et cetera, et cetera. So every single action we make on the world requires us to take into account an understanding of the physics, basic physics of the world around us. And so it's kind of a wide open question of, when we do this all the time, like every action we make, right? So do we have special neural machinery for this? Well, we haven't totally nailed this, but I'm going to skip over this and just tell you that there's ongoing work in our lab suggesting that roughly those purplish bits there are engaged when people do tasks that require them to evaluate basic physical things, like whether a Jenga tower is likely to fall or not, or the predicted trajectory of billiard balls that are moving around and knocking into each other and the walls, and things like that, compared to other kinds of equally difficult tasks you can do that don't require your intuitive understanding of the physics of those things. So it's early days in this work, but it's quite possible that a set of regions are systematically engaged in processing intuitive physics. And these ones, unlike all the other ones I've talked about, are not as selective. These regions do other things. They don't just do intuitive physics. It seems like they're also involved in things like planning action. And we think that's pretty cool, actually, because we think that maybe a big chunk of why we have an intuitive physics processor in our brain is that we need it to plan action. So the idea that those things are all part of the same computational device is potentially really deep and interesting.
Like
Yeah.
In the front part, is this like the supplementary premotor area?
Approximately.
Okay.
Yeah, it's kind of a big ratty bunch of stuff that overlaps lots of other things.
I see.
Yeah,
Okay.
yeah, yeah, but it includes some SMA.
Does it involve like a mirror neuron system as well?
Probably not. Yeah. The most recent bit that we've been studying is pretty cool. So there's lots of bits of brain that I imagine you've heard about from others, not just face processing things, but lots of regions that are involved in different aspects of social perception and social understanding. But all of those things involve your ability to understand or perceive a single other person or agent. This work, we're showing that there's a patch, approximately that little blue guy right there, that is engaged when you watch two individuals interacting with each other. Okay, so this is work from Leila Ishik and Cami Koldewin, and the little patch in question is this little red guy right there, and it responds about twice as strongly when you look at little videos like this, the one on the top. You can see it's an extremely simple interaction, but those agents are interacting with each other in a way that these guys are not. Can you see that? And similarly, that same region responds twice as strongly when you look at an animation like this. Can you see this? This is based on the classic Heider and Simmel ones. You see an agent, it's got a goal, it's trying to get out of there. Here's another agent that's helping it. Isn't that heart-rending? Okay, here's another one. He's trying to get out of the box. Now here comes the other agent, and this one's mean. Okay? So in that little patch of brain, the response is twice as high to the interactions, whether it's the little dots moving around or these shapes compared to physical interacting shapes. And further, if we do this pattern analysis method, we can tell whether the interaction is a helping one or a hindering one, right in that region. Yeah? So we are so out of time. I'm just going to list the other questions of the broad research program. So we just talked about better evidence for the specificity of each of these things, what other ones might exist. Really what we want to know more is not just that a region is specific for function X, but what exactly is represented and computed there. And some of these efforts to use pattern analysis are giving us some clues about that. The methods are not perfect, but they're better than nothing. We want to know how these things arise in development, what their time course is, and fundamentally, what is the role of experience in development of these regions. And there's lots of exciting but sort of conflicting evidence on how these things develop. We want to know about their evolutionary origins. Well, I have 5 more minutes?
15.
Oh, 15.
And then questions.
Oh, all right. I don't know whether to go back up or keep going. I think I'll just keep going, and then you can tell me which of these things you want me to back up to. All right? I thought I had to end on one. All right. We want to know how these things evolve. right? And that's really hard to tell, but here's one little snippet of data. This is data from Rosa Lafer-Souza, who had shown previously that in monkeys, this is a side view of a monkey brain, there's a systematic progression as you go down the temporal lobe of face-selective patches in purple, color-preferring patches in blue, and place-selective regions in green, okay? And so that when Rosa did in my lab is to run similar experiments in humans, and this is the bottom of the brain in humans, so that's the back of the head, front of the head, temporal lobe here, it's kind of hard to see, everybody oriented. And you see the same progression, faces, color, places in humans. And that's pretty weird. Like, why, if you were designing a brain, why would you put color processing machinery between faces and places? I mean, you could, but it's kind of arbitrary. So the fact that we see that totally non-obvious, weird, but very similar pattern in monkeys and humans suggests to us that that structure is inherited from a common ancestor, right? There's not a deep computational reason you could do that, at least not that we know of. So probably that suggests that these structures are homologous, right? Homologous means inherited from a common ancestor, okay? And that's important, one, to tell us something about evolution of brains, right? And two, it's important because as I was lamenting at the beginning of this talk, the methods are so much more powerful in monkeys than humans. And to me, that makes it an even better argument that if you want to understand this stuff, you should look there, unfortunately, right? Where you have more powerful methods. It's a further evidence that monkeys are probably a pretty good model for humans, at least as far as these functions go, right? Okay, here's another really obvious question. You look at that diagram and like it's you know, all these things are, I don't do anything subtle. I just do that. You look at it. How can you not wonder, how does it develop? Why is it organized like that? You know, how does it evolve? But another really obvious thing is, what are the structural connections between these regions, right? As I mentioned, little bits of cortex don't operate in isolation. They talk to other bits of cortex and subcortex and all of that, right? So what are the structural connections between these regions? And shockingly, we don't know the answer to that, like pretty much at all. So there's been all this talk about the Human Connectome Project and millions of dollars flowing all over the place and lots of hoopla and declaring victory, but actually I don't think we know the actual structural connectivity of any of these regions. And that drives me nuts. There are two basic methods to look at structural connectivity in human brains. There's diffusion imaging, where you use a kind of imaging that shows you the direction of maximum diffusion. And it works great for finding big fiber bundles, right? So you have big fiber bundles in the middle of the brain in the white matter, and where there's a whole bunch of fibers going in one direction together. You can totally see that with diffusion imaging. You can see water's diffusing more along the length of the fibers than across. But if you want to know about a little patch of cortex where the fibers that come out of that are small, interleaved with other fibers, crossing other tracks, and doing all kinds of rigmarole like that, I don't think those methods work very well. Yeah, question?
So on that and the multiple voxel pattern analysis, like, has anyone looked at the areas kind of like between V1, like initial visual processing, and the cortex area, and look to see if they can decode at a smaller level, like face perception, the way that they did with like cars and shoes or whatever? Like, is that?
Yeah, so lots of people do that. I'm just giving you little snippets in the field, but there's lots and lots of work like that. So there's pretty good knowledge. You know, V1 projects to V2. This is pretty rock solid. From monkey work, you can sort of see it in humans. V2 probably goes to V3, but now we're starting to get into the squishy zone where it's a little mucky even in monkeys and more mucky in humans. And V4, and then there's like, you know, a bunch of object processing regions, and it's a little, you know, it's a little less defined than that. The V1 and V2 are very clear because each of them has an entire visual field map. And so you can move stuff through the visual field and you can map it out, but you can find its borders and they're rock solid, right? So those ones are really clear. And so you can find those regions, and then you can do multiple voxel pattern analysis in humans in each of those regions separately. People do stuff like that, absolutely. You can say, you know, some things about how those representations change as you go up the system. And it's, you know, a whole lot better than nothing, but a whole lot worse than what you can see if you can actually record from hundreds of neurons in monkeys in those regions. Yeah?
So you talked a
I didn't say easier. No, 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 no. No, I mean, look, doing monkey work is horrible. You have to spend 2 years doing a single experiment. I can do an experiment in a week. So it's not easier, it's much harder.
Yeah.
But the ultimate data you get out has spectacular precision in space and time, and affords lots of causal tests.
In that row, what would be the next best thing if you could brain imaging for you?
The next best method.
Yeah. Like
It's the stuff I showed, the intracranial stuff that I showed you with that patient, by far the best method in human cognitive neuroscience. The trouble is we have no control over it. You know, when I get a phone call through my various connections, which aren't all that fabulous, from a neurosurgeon who's doing something and who's willing to, ask their patient if they mind listening to our stimuli or looking at our pictures, we celebrate because it's so exciting and so much better than any other data we can get. But we can't make it happen, and it doesn't happen very often, and so it's very sparse.
so in terms of non-invasive, do you have like an idea of what you're
Well, I think, functional MRI, as I say, it's gotten us pretty far, right? It's the best we have with spatial resolution non-invasively in humans, clearly, unambiguously. One of the things I skipped over but I'll mention briefly is, looking at time is extremely interesting in the brain. Like if you're interested in computations, right? If the brain is a computational device, which it pretty clearly is, computations unfold over time. And computations involved in things like vision and language unfold over time really fast. So if I flash up a complex real-world image of a complicated scene with all kinds of stuff going on, you have understood the gist of that scene within 1/5 of a second. And that means that the individual computations that make up your ability to extract the gist of that scene happen on the order of 10 or 20 milliseconds. And so we are not even close to being able to resolve that kind of time with functional MRI. Just forget it, don't even try, right? And sentence understanding, like you guys have understood the sentence I'm uttering now before I finish uttering it, right? Because you have predictive methods, you can, it's extremely fast and online. So for things like language and vision and audition, those computations are super fast, and to understand them would be to understand the sequence of processing steps, to understand how they work. And functional MRI will get us exactly nowhere. It'll tell us something about what the different computations might be, right? If a whole patch of, it tells us, if you want to understand visual recognition, you might think differently about face recognition and object recognition. Okay, that's worth knowing. But it won't tell us a sequence of steps. But there are other methods that can give us clues in humans. And something that's relatively new in the last 5 to 8 years is decoding methods with MEG and EEG. Right? So what you can do is you can stick lots of sensors all around the head. They can be just little electrodes you glue onto the scalp, or they can be magnetoencephalography things that are similar, except that they cost $3 million instead of 50 grand. And they don't even work that much better. They're little magnetic detectors that detect the magnetic consequences of neural activity on the surface of the scalp. And in both of those cases, you have beautiful time information and crappy spatial information. But if you're willing to just say, okay, the hell with space, I'll just do time right now, you can ask all kinds of interesting questions. So for example, my postdoc Leila Ishik published a paper a few years ago for her PhD work. I wasn't involved, I'm just a fan of this work, where she used MEG, and she presented objects to people. And what she does is she takes the data from 300 sensors around the head at each time point after stimulus onset. Okay, so time's going this way. I'm sorry, I can't quite do this backwards. Okay, time's going this way. Stimulus onset's here. And at each time point after stimulus onset, you take the 300 sensors, you train a little machine learning thing, and you say, what can I decode from the information that's present at 50 milliseconds, at 100 milliseconds, at 150 milliseconds? Okay, it's just like MVPA, but you do it over sensors across the scalp rather than across voxels within a region. Okay, and what Leila showed is that at 80 milliseconds after stimulus onset, you can decode whether a person is looking at, say, a dog or an apple. Okay, and that's cool, but it's suspicious because it's too fast. 80 milliseconds is about what it takes to get to V1, right? So what Leila then showed is, well, that's true if those stimuli are all of the same size and the same place. But if you have subjects fixate here, and you present the apple and the dog up here, and you train your classifier up there, and then you test it with the apple and the dog down here in a different place and maybe a different size, now you can't decode it until around 140 milliseconds after stimulus onset. And that's exactly what you'd expect. When you get to V1, you have this spatial representation. You can decode all kinds of stuff by how big it is and where it is there. You can tell the dog from the apple right there. But if you need to generalize and have a more invariant representation, that's a lot of what the ventral visual pathway is computing, is those invariant representations of objects. And that takes another 40, 50 milliseconds. So you can see that kind of stuff with MEG. It's super cool. And so now we're having a little infatuation with MEG in my lab right now. And we're doing lots of decoding of stuff across time. Does that mean I'm supposed to stop? No. OK. All right. I'm supposed to stop in a few minutes, though. Is that right? Oh, all right. Great. So for example, I wonder if I have this slide. So I mentioned Leila's work showing that that you can decode, her work with functional MRI showing that region that seems to be involved in perceiving social interactions. Oh, what is the matter with me? I didn't include that slide. That is so dumb. Oh, here, yes, I did. Here it is. Here it is. Here it is. Okay. Okay. So there's that little patch of the superior temporal sulcus that responds more when you look at a social interaction than two people not socially interacting. And sorry, this is a little hard to tell, but here are some of our stimuli. We went around Kendall Square and just, walked into coffee shops and bus stops and took pictures of people who were talking to each other and said, okay, just slightly change so you're not interacting. Or if they weren't interacting, we made them interact. And so we have these very, very similar images where they're either interacting or not, right? And so Leila presented those to subjects while recording MEG responses. And she then asks, at each time point after stimulus onset, can you decode which of those images is presented? And first we just decode, that from that, from that, from that. So it's not decoding social interactions versus not. It's just any information, the scene, anything. And that you can decode at around 80 milliseconds. That's probably V1, okay? But next, what you do is you train on these guys, social interaction versus not, and you test on those guys. Okay, so now this is asking, when do you have abstract information about the presence of a social interaction? And that is just a teeny bit later, but really early, 140 milliseconds. And so what we think that means is that perceiving a social interaction is like recognizing a dog, right? It's a basic perceptual process. It really matters to us. There's special machinery for it. And probably because there's special machinery, we can do it really fast.
And
Yeah.
what are the features of social interaction? For example, like the orientation of people, like looking, like direct gazing or averted gazing. So like what is important?
Yeah, those are all great questions we don't know yet. So we're doing a lot of stuff to look at that now. What I will say is that the first few layers of a deep net trained at that discrimination can't do this. So it's not we're not just decoding like orientations or something stupid like that. But it could still be some sort of mid-level perceptual stuff.
And also the spatial resolution is not that great, right? So
Terrible, yeah.
So we don't really, because of the computational model, we're like inferring that the brain is also doing something similar, right? So you're training a deep net and then you're not seeing representation early parts. But
well, here I'm just using the deep net to say there isn't an obvious visual confound in those stimuli. That's all I'm using it for.
Okay.
Yeah.
But we're inferring that, right? Because of the spatial, because of the time, we're inferring that the later part of the visual cortex are being recruited.
Yeah.
Okay.
Yes. Yes. Exactly. Yeah.
Maybe like a simple question, but is there like a lot of work looking at dysfunctional selectivities in people with particular deficits, like people with autism or other minds?
Yeah. Yeah. There's lots of work like that, and it's pretty frustrating. Mostly, I'll give you two examples. So let me go back to one of those pictures. Most of my slides have it. Okay, let's go here. That's Charlie. He's upstairs. Okay. This little patch here. So I didn't get into this, but it's one of my favorite patches. This is the work primarily of Rebecca Sachs. Other people work on it too, but she did all the early best work on it. And she showed that that region is extremely specifically involved when you think about what another person is thinking. And that's pretty astonishing. That tells you, for example, that we don't just have specialized brain regions for perceptual functions. Some of the most abstract, uniquely human, complex things that we do have specific brain regions, and that's one. So that would be a likely locus for something to be off in autism, right? Because that is a task that people with autism are essentially by definition impaired at, is thinking about what other people are thinking. But oddly, in low functioning people with autism are very hard to test. They don't hold still in the scanner. You can't really scan them. They can't do your task, and so you don't know. So most of the literature is done on high functioning people with autism who are clearly autistic, but can follow instructions, can do your task, can hold still in the scanner. And so Rebecca has scanned a large number of people with autism, and I forget what the number is, but it's quite impressive. And she has looked at that region in high-functioning people with autism and control subjects, and that region is identical in size and location and selectivity and everything you can see with functional MRI. Which is, now this is in people who can do basic theory of mind tasks, right? So people with autism pass theory of mind tasks later, right? Like 8, 9, 10 instead of 4, right? But you're scanning people who can do the task, otherwise there's no point in scanning them, right? And so if they can do the task, they show the identical activations. Now, that doesn't mean that that region is doing the same thing. It means it turns on in the same circumstance when people are doing it. It means, it suggests that people with autism haven't learned to solve theory of mind tasks by a completely different pathway. They're using the same bit of their brain to do it. All of which is very interesting. The thing that she found that I think is just like thrilling and wonderful is that when you use pattern analysis and ask, okay, what's represented in there in people with autism versus typical subjects, she finds a difference. And I forget the exact difference that she finds. I think it has to do with the different pattern you see in there when you read about another person doing an act intentionally versus accidentally. Okay? Which is all about intent. It's all about mental state. You can decode intentional versus accidental actions in that region in typical subjects, not in subjects with autism. That, I believe, is the only clear difference in that region in the brain in subjects with autism, which is pretty shocking, right? Because the behavioral differences are pretty extreme. So that's one example. I mean, it should have worked, just doesn't happen to be true, right? It was a totally sensible hypothesis. Everybody's surprised that that region isn't more obviously globally different, but apparently not. The other example I'll give you is people with developmental prosopagnosia. And so these are people, about 2% of the population, who are really, really, really bad at face recognition. Okay, so the range of abilities in face recognition is surprisingly large. So the people who are in the top 2% are like eerily good. They're so good they hide it because otherwise people find it creepy. Like people who are, they're called super recognizers. There's a really fun New Yorker article about them a couple of years ago. They're being used by crime units in London. And people like this learn that, you're standing in line in a movie, you don't say to the person in front of you, even if you absolutely know it's true, you were sitting next to me in a cafe 5 years ago in such and such, right? You just censor that. You know it's true, you know it will freak them out, you just shut up, right? So there are people who are way up there, and then the bottom 2% are people who routinely fail to recognize family members. I mean, they're really bad. And these are people who are otherwise normal in every other respect. Like they're normal IQ, normal everything, normal reading, normal object recognition. For the most part, there's a little bit of funny business there about some of them have a little bit of some other object stuff going on. But anyway, so it's a very interesting thing. And so wouldn't you think that there would be a big whopping difference right there? I would have thought. And there isn't. So it seemed it might be slightly smaller, slightly less selective. There's some papers that show that. The effect sizes are not enormous. They all have fusiform face areas. And I think what all this shows is that these initial crude findings of regions that are specialized in X, Y, or Z, they're great, they're worth knowing, but they so underrepresent the richness of what's going on in there. The fact that you have a region doesn't at all tell you whether it's working right, whether it's connected up right, all these other things that can go wrong. Sorry, that was a very long answer to your question. Other questions? Yeah. Oh, no, that wasn't.
I was wondering if anyone's trying to figure out if there's like a specialized region for understanding like causality.
Yeah, good question. I think that's going to be in those physicsy regions, but I don't think that's nailed yet. I mean, physics, intuitive physics is sort of ill-defined. There's lots of things that go into intuitive physics. When I last looked at this, which was about 2 years ago, there were 3 papers. None of them were amazing. I forget exactly what they found. I didn't find it very convincing. You have to take causality and think about exactly what you mean by it. There's lots of kinds of causality, lots of ways to implement it. You can do the kind of standard Michotte launching thing like that, versus like Michotte showed, right? Everybody see causal effect of this thing on that thing there, but not in this case, boom, or whatever. If I got the timing right, it would work. And there's some studies that have looked at that. I forget what they find. It wasn't very exciting somehow. Yeah, my guess is, so in a lot of our physics tasks, we use, we basically have billiard balls bumping into each other and bopping around. And so causality is built into that. So yeah, I think, I think there's interesting questions there, and one would have to kind of flesh out exactly what one meant by causality in order to turn it into experiments and think about how it relates to stuff that's been done. But it's worth thinking about. Yeah.
To what degree do you think that we haven't been able to find functionally specific areas in these PFC and other places like that, simply because it's very hard to restrict the inputs to that area? Like maybe there's just a lot of internal dynamics, or it's just very hard to get the appropriate feature space into.
I'm not like the most informed about frontal stuff, but I think what we know from single unit physiology and from lots of neuroimaging studies is that even individual neurons in the frontal lobe can respond to lots and lots of different things depending on the task, right? So we already know that we're in a very different space from face selectivity and music selectivity and that kind of thing, right? So those neurons we know they can do lots of stuff. I mean, it's sort of Earl Miller's lab, that's what they do, right? And lots of other labs. So it may be, but there's lots of things that functional specificity can mean lots of things. It doesn't have to mean just operating on this kind of stimuli, right? So maybe someday people will understand that this particular computation happens in this part of the frontal lobe, and this other computation happens in that other part of the frontal lobe. And then that would be consistent with the fact that those neurons can respond to lots of different things, right? And so people are kind of groping towards those kinds of stories, but last I checked, it's still pretty fuzzy. Yeah?
What do you think has been the difficulty in nailing down intuitive physics? You mentioned a few times that you had
There's no fundamental difficulty, it's just early days. And I just feel like, with the face area, me and dozens of other people have tested the hell out of it. I'm much more confident. It has been subjected to rigorous tests for 20 years. The physics stuff, we just published the first paper a couple of years ago. I can say a little bit more about it. I sort of cut it off because I thought I had to end earlier, so hang on just a second here. So the first paper we did was pretty basic, but encouraging. So what we found is that roughly those regions in 4 subjects respond a whole bunch more if you're looking at this video rotating around if you're deciding which side the tower is most likely to fall on, than if you're deciding are there more yellow or blue blocks. Okay, that was our first contrast. When you do that contrast, you get these regions pretty strongly, right, in pretty much everybody. So that was test number 1. And our second test, I don't have the video here, but these guys are moving around and bouncing off each other and the walls, just in a very inanimate way. And here they're socially interacting. And that's interesting because this one is actually more interesting and more fun. Like there's like chasing and flirting and all that kind of social stuff over here. But in both cases, it's just moving dots. And you get about twice the response in those same regions to the physical thing as the social thing, right? Which immediately raises one of the issues that would arise with causality. Do we mean physical causality, or if this guy's chasing that guy, is that causality too? So that's what I mean by you'd have to say what you mean. Right? And it might just converge on physics or it might not, right? Anyway, so this is nice because these are two totally different kinds of contrasts. That's the kind of thing that starts to increase my confidence that we're onto something. I mean, we got to do a lot more, but that's looking good. Okay, so, and then, I think I have another slide someplace. In ongoing work, Sarah Schwetman in my lab is using pattern analysis to ask what is coded in there. Okay, so all right, okay, so we're doing this with Tenenbaum, who loves this idea of these game engines in the head, and he loves to say, okay, that's the physics engine in the head. And I was like, maybe, maybe not, let's see. But certainly if it's anything like a physics engine in the head, then it should represent abstract physical dimensions, just as physics engines do. And so, and they should represent them abstractly, not just the specifics of exactly what's happening in this situation. And so one of the first things we've been looking at is mass. Right? So you should be able to look at a scenario unfold and ask what the mass of an object is. So this is what Sarah's been doing. And so she scans people while they look at videos like this right here. And the judgment is the object that's going to land in the water, is it heavy or, well, it's labeled right there. Okay, it's light. Here's a light object. Oops, hang on. There. Okay, do you see the little thing drop in there? That's the light object. Here's the heavy object. Big splash. Okay, right? Next row, here's a light object getting blown by the hairdryer. Maybe, no. I don't know where those videos went. Let me see if I can run it like this. Yeah, okay, light object gets blown by the hairdryer. Heavy object doesn't budge. That's nice because the motion energy goes the opposite way from the previous one. One confound we can nail. And so here's the object being dropped on a pillow. Okay, light object doesn't depress the pillow much. Heavy object depresses it a bit more. Okay, so we scan people looking at this, and we ask whether we can decode from those candidate physics regions mass. Okay, and importantly, we ask whether we can decode abstract mass. We do that by, say, training on these two and testing on this one. We do all the permutations of training and testing. So if you have a pattern that represents the difference in heavy and light here, that generalizes to heavy and light here, now you start to be onto something. Because it's not just motion, it's not just this, that, and the other, it's something kind of abstract. And that's the whole idea of a physics engine, is you should have these abstract representations. So that works really well. And various controls work. You can't decode the color out of those regions, even when you're doing a color judgment on the same stimuli. And there are various other controls. So that's looking promising. We're still in the thick of this, trying to make sure it isn't an artifact of something, but it's looking pretty good. So yeah, there's nothing fundamentally hard about it. It's just early days. Yeah?
Do you mean train people?
No, I'm sorry. I just was lapsing into jargon there. I meant when you use this multiple voxel pattern thing, remember the question is, here's a bunch of voxels. Is there a pattern like this for one thing or a pattern like that for something else? And so there's various ways of asking that. One is to use machine learning methods where you show it a bunch of heavy instances and a bunch of light instances, and you say, okay, learn those patterns. And then the way you test it is test it on new patterns. Okay, so what I mean train is you train your machine learning algorithm on the pattern that comes from that part of the brain when you look at heavy and light up here, and you tested on the patterns from the brain for heavy and light down there. Yeah? Yeah.
I'm sorry, my question was not really.
That's okay.
I know there's a huge debate behind this, but has there been studies made on innate actions versus learned actions?
Say more what you mean. Like what?
Like, for example.
And what's the question about innate and learned actions?
If you, has there any study made where a person looks at an action due to instinct versus an action like.
You mean in perceiving someone else doing
yeah.
that action, or doing that action
no,
yourself?
Perceiving it.
Oh God, I don't know. What would you look at? Like, what would be an instinctive action that you would look at? Eating or something.
Maybe, a survival skill, or like giving birth.
But why would that be different? Why would that be different for perception?
Like, would it be like, because like for example, would it be like shown in the memory area of the brain, or would it be like an area in the brain assigned for an instinctive action?
Well, I have no idea, but the way I would think about the hypothesis would be, is that something you need to be able to perceive? Right? So I make this sort of post hoc argument that why does it make sense that there's a whole patch of brain that is apparently selectively involved in perceiving interactions between two other people? Well, one, lots of organisms can do that. Even, interestingly, zebrafish can tell whether two other zebrafish are interacting with each other or not. Go figure. Anyways, it's very widespread. 3-month-old infants can do it. And to me, it makes sense because that's how you tell who's nice and who's not, is how they treat other people, right? And we need to know who's nice and who's not, to know how to get around in the world, right? And so that's an example of, this is all post hoc, but that's just an argument for why, to me, it makes sense to have a brain region specialized for perceiving third-party social interactions. So you'd have to say, why would it make sense to have brain regions that care about whether somebody else's action is innate or learned? I don't know. But you should work on that story if you want to test that, right? Yeah.
This is going back, but can you comment? There's like different reports of like the face area actually also being specific to like things that you have a lot of experience looking at. Like,
Yeah.
say, like,
It was a sensible hypothesis. It just isn't true.
Yes. So is it like?
So here's the thing.
In the lower level?
So here's the thing. So yeah, there's a lot of hoopla about, does a face area respond to anything you have a lot of visual experience with? Well, first of all, that's totally wrong because it doesn't respond to words, and we have heaps of visual experience with words. So then you have to say, well, lots of visual experience discriminating things that are all of the same basic structure, okay? And then that's pretty much only faces, right? So those hypotheses are pretty similar, unless you go find people who have other domains of experience, right? So there have been lots of experiments that have looked at that. And here's what you would need to show. What you need to show is you have to find the face area, replicate, it responds like this to faces, it responds like that to objects, right? Now what you need to do is you need to find people who have some other domain of expertise, say car fanatics, and you need to show that their response to cars is higher than their response to other control objects, like here or something. Okay, that part has been shown. But here's the thing. A car fanatic is really excited when a car picture comes on a screen. Much more excited than they are when, say, a bird picture comes on a screen, right? And so there's a big attentional confound. And the usual way you do that is you find another processing region nearby that cares about shapes and objects and stuff that does not show that effect. And that has never been reported in any paper. And that is just like, that's what you'd have to show. Lots of people have looked, me and a million papers, and not found it. A million other people I know who tell me they found that and they can't publish it because there's like a whole Gestapo who prevents you from publishing it. It's just like, it's become so ludicrous. But nobody has ever shown that, right? And actually, I should say, most papers don't even find this effect, right? Like faces, objects of expertise. 1 or 2 papers have found it. One of them, I think, is a legitimate, nicely done paper. There's a couple others that have a million problems. Many, many papers have looked and not even found that. No paper has found this and shown that it isn't the most obvious confound you'd think of, attention, by looking at another region. So there's just really no evidence for it. Could have been true, just isn't.